Hello everyone, so thanks for being connected and thanks Helmut for putting this together. So this is a joint work with Fabrice Bending and we are both working at INSERM in France, me as a postdoc. Okay, so flow-fast dynamics in neuroscience, as in many physical systems, appear due to the interactions of components with different kinetics. So such dynamics can manifest themselves through complex oscillations like spiking, mixed mode oscillations, bursting, or mixed mode bursting oscillations. So we can observe such complex oscillations basically in many different scales, from microscale to microscale in cells, in neurons. And of course, the mathematical systems that we use to describe such oscillations, to understand them, has components has variables with different time scales. So the time scale separation is in general represented by a small parameter epsilon, and the outcoming dynamics depends on the dynamics and depends on the dimensions of the system, and also the dimensions of the fast and slow variables. So slow-fast dynamical system can be represented basically in two different forms. One is the fast system, the other one is the slow system. So those, as we can see, in the fast system, the time scale parameter epsilon scales the dynamics of the slow variables, whereas in the slow system, epsilon scales the dynamics of the fast variables. Those two forms are equivalent if epsilon is different than 0, but they describe different part of the dynamics when epsilon goes to 0. In the fast system, if you take epsilon to 0, we obtain layer equations where the slow variable appears as a parameter. Whereas in the slow system, epsilon equal to 0 gives us a reduced system. And in that case, the algebraic conditions that we have is the critical manifold, which basically the equilibrium points of the layer equation, and the layer problem provides a basic classification for such systems. So if those equilibrium points are non-hyperbolic, which can happen on the local minima and maxima of the critical manifold, we can have folded singularities, which can induce Canard solutions. So basically, the Canard solutions are the trajectories that runs along the unstable part of the critical manifolds. So they have shown to be determining the excitability threshold in type 2 spiking neurons, also determining the subthreshold oscillations and transition to spikes. Also, Canards determine the transition to burst and spiking phenomena in slow-fast dynamical systems. So the model that we are going to focus in this talk is a neural mass model, which describes the mesoscale average temporal activity, synchronized activity of cortical columns. Such models have been widely used to understand the normal brain activity, also the pathological ones, such as epilepsy, Alzheimer's disease, and Parkinson's disease. So in these models, we have subpopulations of neurons, and the interactions are only the synaptic interactions. Each subpopulation is given by basically two functions. One is a sigmoid function, which converts the synaptic potentials received by that subpopulation to firing rates. And the second function is the H function, which converts the firing rate to postsynaptic potentials. So the parameters of the H function is crucial because they determine basically the postsynaptic potentials outcomes. So one is the gain, synaptic gain, the other one is the synaptic time scale. Then the resulting postsynaptic potentials are multiplied by connectivity coefficients before sending to the next subpopulation. So the model that we consider has basically three subpopulations. One is the excitatory ones, pyramidal cells, and the other ones are the inhibitory ones, with fast kinetics, PV+, and with slow kinetics. So the system variables are the postsynaptic potentials of each subpopulation, synaptic gains, synaptic time constants, and the connectivity parameters. So this model is widely used by Fabrice Bending and his colleagues to model the epileptic dynamics recorded by deep EEG electrodes. And the orange quantity that we see here is the local field potentials. So this is a slow fast system. In fact, we will see it's a 3 times scale slow fast system. And being a slow fast system allows us to model very peculiar dynamics, which is observed before transition to an epileptic seizure. In our manuscript, which is under revision right now, we basically modeled the pre-ictal burst regimes, which have, as you can see on the left columns, spiking behavior, fast spikes followed by a slow discharge. So in this talk, we will decorticate the multiple time scale structure of the model and then see how canards play a role in the appearance or disappearance of this complex bursting behavior after identifying the critical regimes. Okay. So for doing this, first we need to non-dimensionalize or rescale our model because it's a strange one. So we have an 8-dimensional system. Okay. So the first thing is to rescale the time with respect to the fast synaptic time constant, and also with that rescaling, we obtain 2 small parameters, delta and epsilon, which is basically ratio of the time scales, synaptic time scales. After doing that and organizing our equations, we obtain the system written in the fast form. So the first 2 equations, first 2 variables, basically gives us the fast variables. The next 4 is the slow ones, and the final 2 are the super slow ones. So the next time scaling, by scaling time with delta, gives us the slow system, where delta appears on the left-hand side of the equations and epsilon is on the right-hand side. And the next time scaling with epsilon gives us the super slow system. Now both delta and epsilon are on the left-hand side of this 8-dimensional system. Okay, so in the fast system, as we have seen at the beginning, taking delta equal to 0 gives us the fast layer problem and defines basically the equilibrium points of this fast layer problem, defines the critical manifold S. In the slow system, taking epsilon to 0 restricts the dynamics on the critical manifold and also gives us the slow reduced system, which is still a 2-time scale system. Epsilon is at the bottom of the system. Okay. From here, taking epsilon to 0 gives us the slow layer problem with the super slow manifold L0. So our favorite objects, which are the critical manifold and super slow manifold, appear like this when they are projected on this 3-dimensional space of fast, slow, and super slow variables. So the red surface that we see here is the critical manifold, and it is not folded. We also see a red curve. Now, the green surface is the critical manifold. The red curve is a super slow manifold L0, which is folded at 2 points, P1 and P2. So between P1 and P2, L0 is unstable, and it has left stable branch and right stable branch. We can also see a bursting trajectory, bursting orbit in this 3-dimensional space. So basically, the spikes of the burst runs in the fast scale, then it lands, the trajectory lands near the P2 point with super slow time. It jumps to the left stable branch of L0 in the slow time and runs with the super slow time a little bit until it reaches to P1 and gives us the next turn of spikes. Okay, so our interest is the dynamics near P1 and P2, because such points are folded. And to investigate what's happening near them, we need to write down the desingularized slow reduced system. For doing that, we should consider the slow reduced system and apply another time scaling, taking epsilon to 0. So in the 4 algebraic conditions that we see here, indeed are the super slow manifold L0, and the derivative with respect to the fast variable now, well, the slow variable, it is but fast for the system, with respect to V0 gives us the fold set. If the time derivative of L0, and after taking the time derivative of L0 and rescaling the time properly, we can write down the desingularized slow reduced system, which has this form. What we need to do now is to look at the equilibrium points of the desingularized reduced system, because it describes how the slow flow behaves near this fold points. And also the equilibrium points of the singularized reduced system corresponds to the folded singularities. Which equilibrium points? So basically there are 2 equations that can give us the equilibrium points of this desingularized reduced system. One is along the fold set, the partial derivative of m, which is L0 impact, with respect to V0. And the other one is F7. So, but the F7 is the true equilibrium of the original system, so it doesn't induce any folded singularities. Whereas at with the partial derivative of L0 curve with respect to V0 gives us the saddle points, which are indeed the folded singularities of the system. So if we compute the bifurcation diagram of the system with one of the system variable, which is B, the gain of the slow the gain of the slow interneurons, we see that for small values of B, P2 is a folded P2 gives us a folded saddle singularity. For large values of B, P1 gives us a folded saddle singularity. So those are the critical regimes, and where do they appear? So if we compute the bifurcation diagram of the whole system with respect to the same parameter B, in the blue region that is marked here, we obtain the bursting behavior. So the bursting behavior for this parameter set under consideration in this diagram appear between 2 hop points. One is H3, the other one is H4. So we will see that, right? The Canard solutions organize the transitions between harmonic oscillations and the bursting dynamics near H3, hop point H3, and they also organize the transitions between the equilibrium points and the multiple time scale solutions near H4. So if we look at H3 first, H3 gives this sinusoidal harmonic oscillations, okay? As the amplitude of those oscillations increases, basically we can see at the left bottom panel, and they start to interact with one of the critical points, P2. And as we move in the parameter space, the trajectories start to move along the unstable part of L0 curve. As they do it, so they become Canard orbits, basically from harmonic oscillations they pass from harmonic oscillations to Canard oscillations. And as this part along the unstable part of the critical unstable part of L0 increases, the jump that we have towards the right-hand side of the right branch of L0 basically results with spikes. So the number of spikes increases as the trajectory passes more time along this unstable part of the critical manifold, and the spike adding continues until we reach to the maximal canard, which is the 6th orbit, the orange one that we see here. After passing the maximal canard, so we don't have any more spike heading, the system, the trajectory starts to enter in the canards with set regime, then basically to the bursting, stable bursting behavior. So once the system completes its spike heading and the bursting behavior, when if we continue to move along the parameter space with respect to B, I will show here. As B increases, the number of spikes of the bursting solutions start to decrease, and basically they decrease as they pass from one peak to the next, until they reach near H4, and we obtain a reverse Canard explosion in this case, where the trajectory is left with only one spike first, and then they become pure relaxation cycle, and those cycles shrinks and terminate at H4 near P1 critical point. So this completes the whole motion, whole range of dynamics. So this is the type 2 regime, as we can see, but the system for another parameter combination can be also in the type 1 regime. So for such, in such case, we can also observe some Canard trajectories, but this time they are homoclinic type. So we can have small Canards, small homoclinic Canards, or the large homoclinic Canards of bursting type as well. If certain parameters are very small, then we don't have this homoclinic Canards, but still for a certain range of parameters, the Canards appears also for the type 1 excitable regime. Okay. So in this work, basically we analyze the slow fast dynamics of a generic neural mass model. So although it was constructed for studying the epileptic dynamics, this structure, which is the model structure with 3 different subpopulations, is generic enough. So this model can be adapted to many cortical columns. In that sense, it is interesting because in this model we observe the existence of Canard solutions, and they organize the transitions to complex oscillations and also determine the excitability properties. Another important thing is that the Canard solutions in that system construct the boundaries between pathological discharge and the physiological activity. So after defining them, in fact, with Fabrice bending, we asked ourselves if such regimes, such critical transitions, can be considered as new biomarkers. So on the left-hand side, we see simulated signal, and on the right-hand side, we see some ECG recordings from an epileptic patient. So in the simulated signal in type 2 excitable regime, and also during the transitions between harmonic oscillations to bursts, the presence of these Canards regimes gives a narrow band activity. So in the type 2 excitable regime, this narrow band activity is around 3.5 Hertz. In the other case, so it can vary between 4 to 6 Hertz narrow band activity. What is interesting is that during the transition from interictal period to ictal period, as we approach to the seizure, we do observe some narrow band activity, for instance, in the green, in the part that is marked in green and with red, and that narrow band activity has 3.5 Hertz oscillations. So we are now investigating and modeling more appropriately such transitory regimes and trying to understand if Canards exist in local field potential recordings. Okay. So before finalizing, I would like to thank the MathNeuro team and Mathieu Doroche for useful conversations. Thanks for your attention.
Yes, thank you very much, Elif. I have a quick question for you. The Wendling model was originally designed to reproduce epileptic activity. And I was wondering if there is a parameter range in which the system actually gets stuck in this point P2. So rather than moving out of it and producing another burst, which could be interpreted as some kind of spike wave discharge, that it gets stuck there. And then also what are potential parameters that could change this kind of behavior?
So in the Wendling model, the main parameters that they change is the synaptic gains. So A, B, and G in the model. So we are playing with B in that case, which is the synaptic gain of the slow interneurons, the somatostatin positive interneurons. And also we are playing with the synaptic connectivity parameters, the connectivity parameters C. The parameter that we are changing in this study basically is the projection of the excitator neurons on the slow interneurons again. So the model has lots of parameters by targeting the most physiological ones, let's say, because it's a biophysiological model, the Wendling model. We can change the patterns or move from one to the next, let's say, yeah.
Okay, thank you very much.